In the pharmaceutical industry, the UV spectrometry and HPLC technique are the most widely used analytical techniques. These are used not only for the APIs but also for the chemicals, but also for the excipients, raw materials, and the finished formulations. I welcome all the viewers to this video on understanding regarding UV and HPLC techniques. Basically, we will understand the differences between these two techniques. These are the widely used techniques in the pharmaceutical analysis. The principle of UV and HPLC. So, by UV, we measure the UV absorbance at a specific wavelength. It is called as lambda max, and we need to find out the specific wavelength, that is lambda max, at which the maximum absorptions happens. And principle of Beer-Lambert's law is followed by the UV. While in HPLC, it separates compounds based on the distribution between stationary phase and the mobile phase. And detection can be done by UV fluorescence, RI, MS, DDA detectors or techniques. Sample requirements for UV. It is best to use clear single component solutions, and HPLC can handle the complex mixtures like drug plus impurities and excipients because there is a column in which the separation occurs. Then UV accuracy affected if the excipients are there, impurities are there which can absorb at the same wavelength. But in HPLC, the separation occurs, ensuring the accurate quantification of the analyte. Specificity and sensitivity. So UV has a low specificity and moderate sensitivity, while the HPLC has high specificity and higher sensitivity. So if the HPLC is the method, then it is highly sensitive than the UV method. Then applications. UV is used for routine drug assay like single API, dissolution testing like simple cases, QC raw material identification, and quick sample screening. HPLC used for the assay and impurity profiling for the pharmaceuticals, and HPLC is considered as a gold standard for the analysis. Then stability studies, degradation products, impurities can be separated and quantified by HPLC. HPLC is also used in the pre-case studies and in bioequivalence and multi-component formulations analysis. Generally, nowadays HPLC is widely being used than the UV for the regulatory submissions to the authorities like FDA, EMA, Brazil, Anvisa, and many more. Then considering the cost and regulatory acceptance, so UV is time and cost effective method of analysis. The UV is fast and cost is low. Regulatory acceptance for UV is limited. It is only or can be used for simple and interference free cases, while HPLC requires much time. It is a slower technique. Generally 10 to 60 minutes per run is required, and the cost is high because it utilizes the columns for separation, solvents, and it requires maintenance. Then regulatory acceptance is more for the HPLC. It is considered and accepted as per the USP, EP, BP, JP, and many other pharmacopoeias for the assays, impurity detection, and dissolution testing. UV is a quick, simple, inexpensive method, but it has limited specificity and limited acceptance, while HPLC is a costly technique. It is highly sensitive, highly specific, and a gold standard for the regulatory requirements and meeting the regulatory compliance. HPLC technique is higher in cost and it requires more time compared to UV. This was the comparative understanding for the HPLC and UV. Thank you for watching the video.